All right, it looks like we're starting to level off on people coming on in, so I will go ahead and get us started. Thank you so much for joining this session of our journal club. As you can see on the screen, we're going to be talking about this recent paper from our colleague Jonas Bergquist, who runs our collaborative center at Uppsala in Sweden. And the paper is Plasma Proteomic Profile Reveals Persistent Immune Activation and Post-Acute Sequelae of SARS-CoV-2 Infection. Really quickly, before we get into, the meat and potatoes of the paper, I just want to go over some housekeeping items. If you were able to join our last journal club session, this is basically a repeat, and I'll probably do this basically every time we do a journal club. So apologize if you hear this more than one time, but just a reminder, you can go ahead and put questions in the Q&A channel as we go along. I will do my best to try to see questions as I go, but I'll definitely try to answer them at the end if I miss them as I'm talking about things. I want to give some quick context for the perspective that I'm bringing to the conversation. So I'll say upfront that I am a researcher, not a medical doctor. Part of, my scientist training is to really be cautious about claims that you make based on the data you have available. I will do my best to extend beyond just giving you simple results and try to draw some connections to other research, talk about potential implications for people, with long COVID, given the topic of this paper, but also extend that to ME/CFS as well. And I do have to make it clear, anything that I say in the realm of treatment still needs to be thoroughly tested, and I cannot provide medical advice. So I have to say that up front. I'm not going to go over supplemental information today because there's plenty to talk about just within the main body of the paper. And this is really my personal way of explaining things as I'm going through scientific papers. So I'm trying to keep this a little informal to give you a little bit of insight into how I kind of think through the data as we go along. But, like I said, I'll try to answer questions as I can, and hopefully we'll have a decent understanding of things at the end of this. So let's give a little context for the paper. So this is, I'm just saying it's an abstract, but really it's just, some of the basics for what's involved in this paper. To start out with, who are we looking at? We have 92 participants with post-acute sequelae of SARS-CoV-2 infection. So this is PASC, but also known as long COVID. I'm going to call it long COVID throughout the rest of the time just because it's easy. And on average, this was about 34 months after the acute infection. So it's a decently long time afterward. And then there were 73 controls that were matched to the participants by age and sex. And these got COVID around the same time as the participants they're matched to, but they recovered fully with no complications. So they're considered control participants. And then the basic concept of what we're looking at in this paper, the team measured protein levels in the blood, and they found differences between people with long COVID and the controls that recovered from their acute infection. So that's kind of the baseline for where we're starting today. I want to quickly throw up some of the key takeaways at the start, and I'll try to keep referencing back to them as we go through, and then, bring them back up at the end so that we can be reminded of, what are some of the main points from the paper. But just as our general broad overall conclusion, we're seeing consistent low-grade inflammation in long COVID. Some more specific key takeaways, they identified 5 proteins that were considered strong discriminators between patients and controls. They found persistent activation of innate and adaptive immunity. And then there was no difference in spike protein levels between the patients and controls. The only other key concept or term that I want to elaborate on really quickly now is, just because it's one of these key takeaways, what's innate and adaptive immunity? Innate immunity is this general rapid response to any antigen, which is just a foreign substance. So you can kind of think of this as like a first line of defense. And then adaptive immunity is more targeted. It's a learned response to specific antigens. And this adaptive immunity really relates to something that you've been previously exposed to, and actually vaccines kind of are trying to take advantage of adaptive immunity by presenting specific antigens to the body so that you have this learned response if you were to then see that infection in the future. So that's what the difference is between innate and adaptive. General first line of defense versus targeted learned response. All right, so let's go ahead and get into the results of the paper. I'm not going to spend too much time on the clinical characteristics here, but just give you a quick run through of this. So like I mentioned at the start, they matched the controls to the patients, and you can see the age, the average age was pretty well matched between these, so 44.4 and 44.5. The age range in general was 21 to 64 years. You can also see in both sets they're majority females, unsurprisingly. And then there were similar comorbidities across the groups, with the team having mentioned an exception where there was a higher incidence of ADHD in patients compared to controls. And then here I'm just popping up a little box around the most debilitating symptoms as they were reported. These were cognitive fatigue, physical fatigue, headache, and muscle pain or myalgia. And just to note, obviously, there have been many variants of COVID at this point. They found no significant differences in the symptom profile based on the COVID variant. So that's not really coming into play very much here. All right, this table is looking at differentially expressed proteins. So protein expression is essentially the protein level in the blood. So differentially expressed proteins means that there are proteins that are at different levels in one group compared to another. So in this case, the team analyzed the proteins in the blood of the participants and found 358 proteins in all samples based on the method that they were using. They used a statistical test called a t-test to compare the levels then of each protein in the patients and controls, and they found 26 of them that were significant after some mathematical adjustments were made that's necessary for these kinds of tests. And then those 26 are the ones that are shown in this table. So 23 of these are upregulated, so they're at higher levels in patients than in controls. And then 3 are downregulated, so they are at lower levels in patients than controls. And I'm throwing arrows up here just to indicate the 3 that are downregulated, so you don't have to search around to pick them out. And then the rest, like I said, are upregulated. All right, I'm going to go a little bit out of order from what's in the paper itself here because I want to talk about the last column in this table. It's the one that's labeled RF rank. And since it's listed here in this table, I want to switch to the figure that kind of produces these numbers and then come back to this. So this is figure 2, and it's looking at ranked importance values. So what this is the results of a random forest analysis. And that is just a machine learning tool that helps to identify connections between protein levels and groups. So in this case, the protein levels and then whether they're a patient or a control. And it does account for protein interactions that aren't like on a 1-to-1 ratio. They're not linear. And you can, the output of this is really identifying the relative importance of each protein. So it's looking at all of the proteins measured and ranking how important they are for whether someone would fall in the patient or the control group based on that protein. So part A is the outside box, and this is looking at all 358 proteins that were measured. Obviously, that's a lot to look at. Honestly, don't bother. You can't read it. There's just too many to look at. So the insert, part B, is really what I want to look at here. So this is just the top 50 proteins. So as you look across the horizontal axis, it's called the variable importance. So basically, each point in the row, the further to the right, the more important that protein is in the ranking. So you can see the top protein is IL-1RN, and that dot is the farthest to the right, which means it's the most important. And really, the only other takeaway I want to point out here is that for the most part, the importance corresponds with statistical significance. So in this case, the proteins that are statistically significant are noted in red. And you can see for the most part, the more of the red proteins are up at the top, so they're more important and they are more statistically significant. And so I'm bringing it back to kind of the key takeaways that we started at the beginning. This is where we can say that they've identified 5 proteins that are considered strong discriminators between patients and controls. And so going back to the previous table, again, I said I'm going a little out of order here, but I want to highlight then within this table that the 5 that are strong discriminators between patients and controls based on that random forest analysis are the ones that I have in boxes here. And I'll try to keep pointing out these 5 as we go along, just so you can kind of keep looking at them. Okay. Now to figure 1, I'm going to put this up in pieces and then we'll look at the figure as a whole together after we've gone through each piece. So this is part A, which is called a heat map. And I'm going to try to step through this slowly. So all the statistically significant proteins, so the 26 proteins, are listed along the bottom of the map. So that's where the box is right now. Now, within this new box, each row represents one participant. And then on the left-hand side, you have this bar where the orange section represents the participants that are controls. And then the blue section, all of the rows in that section, are the ones that are the patients. And then these lines along the top are kind of showing some of the interactions between the proteins that are listed on the bottom. I'm not going to spend really any time on that because there's a different way of visualizing this later on in the paper, but I just wanted to point it out since it's in the figure. This gradient on the right-hand side shows the magnitude of the up or down regulation of the protein. So the deeper the color, the higher the magnitude. Deeper orange or red is upregulated. Deeper blue is downregulated. All right. So the 3 proteins on the left are the ones that were found to be downregulated in the patients compared to the controls. So I'm going to pop up a box here. These are the 3 proteins, and then I've highlighted just the ones in the patient group. So this bottom section, you can see within the box, in general, it's not the case for every single one, but in general, these are showing up as blue. And that's where you can kind of visually see that these are generally downregulated in patients compared to controls. And then I'll pop up this other box for the rest of the 23 proteins that are considered upregulated. And you can see that in general, again, the patient group is showing red within this area compared to the blue in the controls above them. So this is more just a visual way of seeing the differential expression like we talked about in the previous table. All right, this is another visual representation of this. Figure 1 in general is visual representations of this differential expression. So this is a type of a scatter plot that's called a volcano plot. The vertical axis, the y-axis, is essentially showing the p-value, so that's statistical significance. And then the x-axis, or the horizontal axis, shows whether it is up or down regulated. So there's a 0.0 in the middle of the horizontal axis. To the left of that is things that are down regulated. To the right are things that are up regulated. And then each dot represents one protein. Now, there's a dotted horizontal line across here as well, and that's a p-value threshold. But importantly, that is an unadjusted p-value. So I kind of alluded to this a little bit where we have to do some mathematical adjustments because we are doing a number of mathematical tests. So it's called an adjusted p-value. So the adjusted p-values that are still significant are the ones that are in the orange or red color. Okay, so those are the orange or red dots are the 26 proteins we've already talked about. Now, again, just to kind of highlight some of the differences. So these 3 that I have circled are the three that are the downregulated proteins. So if you were to imagine a vertical line up from zero, these three are the ones that are significant and to the left of that zero line. And then I'm popping up arrows here just to highlight the 5 most important proteins based on that random forest analysis that we talked about. And just an observation that I'll point out here, the 5 important ones tend to be higher up on the graph compared to even other statistically significant ones. So again, in general, like the importance of their ranking is kind of corresponding with how statistically significant they are. So that's nice, kind of validation within based on different computation methods to see that. And here are box and whisker plots. So I'm going to walk through these a little bit, and I know this is a little overwhelming to look at, so I'll point out some major observations, but you don't have to take too much time focusing on this. When you look at a box and whisker plot, there is a horizontal line within each box that is considered the median or the middle value of all the data within that group contains. The box shows kind of the middle 50% of the data, and then the vertical line extends to the minimum and maximum values, excluding any outlier points. And so you can see some dots on the graph. Those are just outlier points. Okay, and then you have all 26 proteins listed along the bottom, and each of those have 2 boxes, one showing the levels for the controls in orange, and then one showing the patients in blue. And again, I've added arrows just to indicate the 5 important proteins. Okay, the only thing I'll say is I would observe from this that the 5 that have the arrows, you can see kind of a more pronounced separation of the boxes on those 5 compared to some of the others. So again, that is, excuse me, kind of playing into the statistical significance. Where there's greater separation, you're going to have more significant p-values. So now putting all of these 3 together, this is all of figure 1 in one place. We can really see that we've identified patterns in people with long COVID that are different from controls. And, we have the proteins now that are significantly different mathematically. So the next step is really to kind of look into the biology behind that. And in order to do that, we're going to talk about gene set enrichment analysis. So this is a type of computational analysis that shows whether a predefined set of genes, so those in a signaling pathway, for example, are statistically significant between the 2 groups. So instead of looking at individual proteins, you're looking at groups of proteins. And this study found 5 enriched gene sets. And so these 5 contained at least 3 genes where the proteins were found to be significant in the previous analysis. And the p-value of the gene set analysis met a specific significance threshold. So this study is showing these pathways listed here as being upregulated in people with long COVID compared to controls. All right, I'll just kind of briefly talk through what each of these might indicate. So allograft rejection, this is related to activation of T cells and the adaptive immune system. So again, really quickly, innate versus adaptive. Innate is your generalized first response, immune response. And then adaptive is your learned, more specific response. The inflammatory response group, probably self-explanatory, it's sustained inflammatory signaling. TNF alpha signaling via NF kappa B. This is related to activation of innate immune system and inflammatory responses. And the same thing goes for IL-6, JAK-STAT3 signaling, activation of innate immune system and inflammatory responses. And then the IL-2 STAT5 signaling is similar to allograft rejection, so it's related to the activation of T cells and the adaptive immune response. So what we can kind of take away from this, bringing again one of our key takeaways from the beginning, is that we're seeing persistent activation of innate and adaptive immunity. All right. Now this is a network that was produced from string analysis. So this is another computational technique that produces a network like this. So it shows how the proteins are interacting with each other. And I kind of alluded to this a little bit in the heat map back in figure 1 with the connections that looked kind of like a hierarchy tree at the top of that heat map. But I think this is, a little bit easier to look at. Obviously there's still a lot of connections drawn here, but we'll walk through this a little bit. So the proteins that were listed in the inflammatory response gene set from the last table are actually circled in blue within this network. And then the color gradient, so the circles, are the proteins and how deep of the color they are. So the more red they are, the more statistically significant they were. So that just relates to their p-value again. And I'm popping up an arrow on the bottom right here just to look at OSM for an example. So this is one of the top 5 proteins that we have seen in the past slides. You can see it's really dark red, so it's more statistically significant in general. It's also circled in blue, so it is part of the inflammatory response gene set. And then you can see connections to other proteins drawn with the gray lines. So I'm not going to spend too much time on this. Just note that obviously there are a lot of protein-protein interactions going on. So that's partially why a random forest analysis is good for determining the important proteins. And then it's also why gene set enrichment is really informative, because it looks at pathways and functions rather than just individual proteins, because obviously there's a lot of things going on here in the biology. All right, last figure. This is looking at the spike protein level. So this is another box and whisker plot. You saw this in figure 1, but just to remind you, the horizontal line in the box is the median or middle value. The box shows the middle 50% of the data. Vertical line extends to the minimum and maximum, and then there are outlier points. So the controls are on the left in orange, patients on the right in blue. And then really what we're seeing is that there's no difference mathematically between the patients and the controls. So at the top, you can see there's a p-value of 0.29. That's well above the significance threshold. And you can kind of see this visually as well. There's a similar median value, similar spread of the box above and below that median line, and similar deviations and outlier points. So again, this points to one of our key takeaways. There's no difference in spike protein levels between patients and controls. All right. So now I'm just going to recap our key takeaways. I'm going to swap the order that I originally had in the first slide, just because I think these 3 more specific points kind of lead to our main conclusion. So these other key takeaways: of the 26 proteins that were expressed differently between patients and controls, there were 5 that were identified as strong discriminators between the groups. The proteins that were different belonged to gene sets that indicate there is persistent activation of innate and adaptive immunity. And then there was no difference in the spike protein levels between patients and controls. So altogether, this is telling us that there is some consistent low-grade inflammation in long COVID. All right. So now I want to try to get to the bigger picture here. So the first thing I want to point out is about 85% of long COVID participants in this study also matched ME/CFS criteria. So I think there's certainly plenty that we can still learn in the ME/CFS space from this paper, even though it is focused on long COVID. And I'll point some of that out in some of the other things that are on this slide, but I just wanted to note that so that we can keep that in mind as we're talking about, relating this to other research. And the results in this paper, I want to point out, do directly complement the results in another paper from Dr. Bergquist's group that looked at the transcriptome analysis of peripheral blood mononuclear cells, so PBMCs, also in long COVID. So this study we're talking about today found no difference in spike protein levels in the blood between the long COVID and the controls. And then the PBMC study found no difference in the SARS-CoV-2 gene fragments in the PBMCs. So these are kind of both suggesting that the inflammation signals that are occurring are independent of any potential viral persistence. And I'm seeing that there's a question kind of asking about this, so this is a great tee up. Because we're not seeing that statistical difference does not necessarily discount viral persistence as a theory. What it really is suggesting is that if viral persistence is causing inflammatory signals, the reservoir of that spike protein or whatever is persisting just isn't found in the PBMCs or the blood. So it could be in specific tissues. There might be some other explanation, or potentially they are independent from each other. But just from the data that we have, the inflammation signal is not necessarily tied to viral persistence. Okay. The other thing I want to point out, just as I have brought up that PBMC paper, both of these studies also found just general indications of chronic inflammation, and then more specifically identified the JAK-STAT signaling pathway as being more activated in people with long COVID than in controls. And in this paper, that's coming from the gene set enrichment analysis. Okay, so the rest of what I have on this slide is trying to summarize some occasions of the individual proteins that we mentioned as, the 5 important ones, and them showing up in other literature. I don't consider this an exhaustive list, but it does kind of highlight that these are showing up in other studies. So this is not, the only instance that we've seen these being different in long COVID or ME/CFS. And you'll see there are a few words here that are in italics. So that's just actually discrepancy between literature and what we're seeing in this paper. And I'll kind of address that when we get to it. So starting with OSM, there was a systematic review that actually highlighted this as one of the most consistent proteins found to be elevated in long COVID. I also want to note in an ME/CFS population, this is one of the signaling pathways that was enriched by some microRNAs that were found to be different in ME/CFS. Looking at IL-1RN, this has been found to be correlated with severity of COVID, and it was also found elevated in a different cohort that was evaluated at 8 months post-infection. So again, we're looking at, on average, it was, I think, 34 months after infection in this study. So, obviously 2 pretty different time frames, but still seeing IL-1RN showing up in both of those occasions. And then in general, the elevation of this one has been correlated with fatigue, sadness, autonomic symptoms, and post-exertional malaise. So, there's some connections to ME/CFS through some of those symptoms as well. HLA-DRA plays a role in eliminating viral infections. So it's possible that this could be linked to some of the theories on viral or antigen persistence, but it was also identified in a long COVID cohort through immune profiling. So, just another way of identifying different proteins that are different in that population. So having it come from, even a different method is, I think, reassuring. And then in the ME/CFS space, alleles, so variations in the HLA gene, are also associated with ME/CFS. So there's that connection as well. Then looking at ANGPTL2, it's been correlated with cardiorespiratory fatigue and anxiety depression symptoms in a separate long COVID cohort. But then in a different cohort, it was actually found to be decreased. And you might remember in this specific study, it was elevated. So there is a slight discrepancy amongst the literature in that sense. And kind of similarly for CLE-C4A, it was downregulated in this study that we're talking about today, but upregulated in a study comparing long COVID to controls that was separate from this. So the one thing I want to note about these discrepancies, it's possible that it could be explained by different subgroups. It could relate to disease duration. There are plenty of explanations for why they might be different, but I think what's important to take away is that ultimately it still means that the pathways that they're involved in are dysregulated in some way. So, the fact that they're still showing up in other cohorts, even if they're not in the same direction, I think still has some significance to it. All right, next up, I'm going to actually kind of wrap up the discussion with research into treatments in this area. So before I pop up this slide, I just want to reiterate that this should not be considered medical advice. This is pointing out treatments that might merit further research in long COVID and ME/CFS, purely based on their mechanism of action and how it relates to the findings of this paper. So just keep that in mind as I'm walking through this. I have these grouped by pathway. So thinking about TNF alpha and NF kappa B, there are anti-TNF biologics, so there are a few listed on the side here. In general, these suppress TNF alpha, and then downstream of that, so within the signaling pathway further down the pathway, NF kappa B, and these are used in autoimmune diseases. In ME/CFS and long COVID specifically, there's really only anecdotal reports of using some of these and no really replicated trials showing clear benefits. So again, this would really need further research. And there is, of course, an increased risk of infection when you are suppressing some of your immune pathways. So again, something that, has to be considered very carefully. And then within this pathway section, there's also indirect NF-kappa B inhibitors and NRF2 activators. And some of these are listed out here. And the concept of this is really to reduce chronic low-grade inflammation and oxidative stress that generally tries to maintain NF-kappa B activation. Metformin is listed here, and that's kind of been one of the more studied treatments within ME/CFS or more commonly talked about, but it's still really only with smaller, pilot studies. So there's nothing definitive to say on that yet, or there needs to be still more understanding of who it benefits and why. Then within the IL-6 and JAK-STAT3 pathway, anti-IL-6 or IL-6R antibodies are listed here. So these generally block IL-6 signaling and reduce STAT3 activation, again, downstream of that, and, generally block inflammatory gene expression within this pathway. There's not a randomized trial, again, to point to within ME/CFS or long COVID, so there needs to be a lot more testing in this area. And similarly for more broad JAK inhibitors that are listed on the slide, in long COVID, there is, I believe, a trial looking at baricitinib and related agents. And, this has been proposed for treating persistent inflammation in long COVID, but again, robust data isn't there yet. So it's still in progress. And again, with these, there are certainly risks involved. So more controlled research is needed on it. There's some question about whether long-term use for ME/CFS and long COVID is appropriate for some of these because of how, I guess, kind of intense that they are. So I want to throw some extra caution in there on here. IL-2 and STAT5 and T-cell regulation. So there are some immunosuppressants that are listed on the slide. So These generally reduce IL-2 production or T cell proliferation and kind of broadly dampen the adaptive immune system. And I think in general, these have been more used when there's a comorbidity of autoimmune disease involved, but they're not established as treatments for ME/CFS or long COVID and do obviously, again, carry a lot of risk because they're kind of more blanket suppression of the adaptive immune system. So there's certainly risk involved with that. The IL-1 access and IL-1RN context. IL-1 blockers are listed on the slide. So they inhibit IL-1 signaling. And once again, not a lot of evidence in ME/CFS and long COVID specifically, but I would imagine that this would really come to play in precision medicine style approaches with, wanting to see IL-1 signaling specifically within the patient that's being discussed for this treatment. But again, would need some more testing before that can be really thoroughly talked about. And then just in an other relevant or emerging approach kind of sense, we have microglial neuroinflammation targeting agents that are listed here. So these aim to kind of dampen the central nervous system inflammation and glial activation, which can be downstream of cytokine or JAK and NF-kappa B signaling. You'll note that low-dose naltrexone is actually listed on here as one of these agents. So I will just mention that, OMF has a clinical trial right now that's looking at low-dose naltrexone and, who it might be benefiting and why. So hopefully we'll be able to provide a little bit more information on that when that trial has concluded. Okay, so I was able to answer a few questions along the way just as I was talking. But one other that was submitted ahead of time that I want to make sure I address is just asked if there are plans to distribute accessible language materials for this particular research to engage the broader ME and long COVID community. So OMF does put out social media posts for each of its publications with just really short, high-level summaries. So if you don't follow us on social media and you want to be able to see some of that, that is certainly something that we do as well. So we have a question in the Q&A that asked if the authors talk about the suitability of Olink PEA plasma protein analysis for use as a diagnostic test. So the random forest analysis that they did was specifically designed to get at these, ranked importance as opposed to developing an algorithm that could be used for a diagnostic. So with this particular study, didn't have that as an objective. It's certainly something that could be considered in the future, but would need, probably a higher sample size. And I think you would want to include other control populations as well to really get at a true diagnostic. So it could potentially move that direction in the future, but that was not the original objective for this specific paper. All right, those are all the questions that I have so far. I will keep things open for a minute if you're trying to type something. All right. Is there any segregation of the long COVID patients that were experiencing PEM? As a long COVID patient, I believe something is released or elevated during PEM. First, I'm sorry to hear that you're a long COVID patient. I hope you are able to find some relief. There was no specific analysis of post-exertional malaise in this particular study. I will say I think this was in some of the supplemental information. They did do some quick analysis on some of the proteins to see if the levels corresponded with some of the variables in the clinical picture, and they didn't find anything to be statistically significant in that sense. So kind of in general, the protein levels were independent of the symptom profile, but I don't know specifically about PEM in this case. And there was no, induction of PEM as, part of the study. So in theory, the proteins that they were looking at were at baseline levels for these people. All right. I'll leave it open for another minute just in case there's anything else trickling in. But yeah, we have a comment here. It'd be interesting to identify if patients were in a PEM state or in general symptom flare-up at the time of blood collection. Absolutely. Again, the hope is that they were just at a general symptom flare-up at the time of blood collection. But of course, coming into a clinic, maybe there was already some PEM activation going on. We just don't know based on the information that we got today or got from the study. And yes, there will be a recording shared with anybody that was registered for the session. So you will be able to go back and look at things as you want to, or if you missed a section, you can always go back. Are the proteins identified significant in terms of the symptoms of long COVID? So the proteins were identified as significant between the long COVID patients and the patients that got COVID but recovered. So they're not necessarily correlated to specific symptoms, but based on, a definition of long COVID. And again, I think, 85% of them also matched ME/CFS criteria. So there's a lot of overlap going on there. But from what they found, at least the proteins were not necessarily mapped to any specific symptom or severity of symptom, but rather long COVID as a whole compared to the control group. All right, I will wrap us up there then. Thank you all so much for joining. Hopefully, you got a little bit further insight into the paper from this. And as I think I close things out, you'll probably be taken to just a really short feedback survey. If you have anything to share that for the session, I am happy to take your feedback. Thank you all so much for joining, and I hope you have as good of a day as you can.